interference. This lecture is officially scheduled for 2 hours. I won't be filling those 2 hours, so a bit spare time left. I do think it's wise to plan a break in between. So maybe after 45 minutes or a bit longer, I will send you off for 10 minutes. Then we can return. And the most practical way to do it is first to give you the theoretical background, of course, and some practical tools of the use of RNAi in current research. And then probably after the break, I will be telling you something about my own research, which is actively developing at the Department of Cardiology. And I use a lot of the techniques that we will be discussing in the first hour. I use them in the lab to approach the development of the pathway and to study the development pathway. So the latter is, of course, not for the exam. Something that you may find interesting because it's a translation of the techniques that you will be learning about into a more practical daily use. I think that's very, very insightful for you when you want to become a researcher in the end, that you already know how researchers think and how they use the theory actually that you are being taught about in the studies. Alright, so my name is Klaus Schoen. I'm a university lecturer at the Department of Cardiology. My background is I've studied biological health sciences here in Maastricht. I did a PhD in cardiology as well, and now I'm officially a molecular biologist. Let's call it that. With a special interest in RNA mechanisms. So as I said, the first part of this lecture will be on mechanisms and, of course, the role of RNAi and siRNAs in health and disease. And secondly, I will tell you more about how I use this knowledge in my daily work. Very basic definition of RNAi is, you can type it in Google, that's what I did, and you will find a lot of definitions, but it's actually quite simple. It's a process within living cells that moderates the activity of the genes within these cells. That's what you find in Wikipedia, which is actually a very, very nice background tool, as you may probably know. In medical dictionaries, so more for doctors, they say it's a process in which the introduction of double-stranded RNA into a cell inhibits the expression of genes. So that's also more directed towards, or more thought towards a therapeutic use as well, in the enteral food. It's a very recent discovery. You will probably also have read it in my handouts that I already put on or will do. In the 1990s, the discovery of microRNAs was actually the first discovery of small RNA molecules that play quite a central role in the regulation of gene expression. But people did not really understand what it meant. So in 1993, Lin4 was discovered in C. elegans, so that's a little worm. And nobody really thought that also in humans, these small RNA molecules would play important roles, but we know better now. In the meantime, lots of processes have been discovered. The term RNA interference was introduced in 1997. Then we've been discovering that in the years after, that RNA interference can be very broad. That, for example, mammals have microRNAs, but plants also have microRNAs, which are small endogenous RNA molecules, so encoded from normal genes, but I'll come back to that. And these microRNAs regulate the expression of many, many genes in the same organisms. On the other hand, you have a small interfering RNA, which is also, on the one hand, part of the endogenous mechanism of especially plants, but also of bigger animals, bigger organisms. But they actually can be introduced by viruses as well. Viruses use RNA interference as a way to modulate host function, and to enhance, of course, their infectivity. In the end, so we have now probably reached, with the human genome sequencing project, we've reached the end of the discovery phase. Now, while we before thought that most of the genome was not coded, so only 1% of the genome actually codes for protein coding genes, we now know that most of the genome is coded and is transferred into RNA, transcribed into RNA, but most of these RNAs are non-coding and somehow influence protein expression. On the one hand, by RNA interference, so targeting already transcribed messenger RNAs and preventing them from being translated into protein. On the other hand, by other epigenetic mechanisms, for example, targeting chromosomes and mediating or modulating methylation, the binding of transcription factors, those kinds of things. I will be mostly focusing on the RNAi part, so on the post-transcriptional modification of gene expression in this section. As you see, RNAi is becoming really, really popular. So in 2009, there were already almost 5,000 publications in this field, so in the non-coding RNA field, which is also including PRNAs, or PV RNAs, that are actually part of the gonads, so in the chromosomes. And the regulation of these sex chromosomes, CRNAs, MRNAs, and recently the long non-coding RNAs, which I will give you one slide at the end of this lecture. So to start with the theoretical part, I will be addressing all of these blue squared topics. So where do they come from? The RNA interfering molecules, endogenous, exogenous types. So already talked about microRNAs, which also have the small hairpin RNAs, for example. The mechanisms, the mechanisms that they use to regulate post-transcriptional gene expression, and the key roles they have in learning. First, starting with the types. I think that's the most logical order to do it. Actually, the most prominent ones that are now now recognized are microRNAs, and you can compare them directly to siRNAs, small interfering RNAs, which are both small non-coding RNAs, 21 to 25 nucleotides in length. But on the one hand, microRNAs are encoded for normal genes, as I already said, and at the moment, I think more than 1,000 already are known in the human genome. And this is the way they are encoded. So there's lots of possibilities, and we don't know half of the function yet. It's really a developing field. You have certain types of genes, intergenic polycystonic MRNAs, as it's called. These are genes that do not encode for anything else but the microRNAs that are inside them. And these microRNAs are in a cluster together. Often they have one promoter, and often they have common functions. For example, it can be anti-metastatic, AIDS, it can be. No. I'm a little bit out of breath because I'm pregnant, I'm sorry. So I'm grasping my breath. But I'll try to hold it through to the rest of the Then the other group of RNAs is the intergenic single microRNAs. It's very simple, it's one microRNA encoded by one gene. There's nothing special about that. The exonic microRNAs in the non-coding gene, an example of that is microRNA 155, which is encoded in the non-coding BIC gene. BIC is a gene that was already discovered in the 80s, for also being involved in metastasis of non-small cell lung carcinoma. So this gene has already a very large history and was early discovered as being important in the development of cancer. But nobody really understood what it did. And only recently people found out that actually it's the microRNA inside this gene that has the most potent function in this type of cancer. I'll be talking more about 155 when I give a practical example of how I use RNA interference in the lab. Because microRNA 155 is one of my primary targets as well, but then in relation to half. And there is the intronic microRNAs in the non-coding gene, and then finally you have intronic microRNAs in the protein coding gene. And actually the latter group is quite interesting because these microRNAs are often found to have or be part of some feedback loop to regulate the expression of the host gene. That's a very well-known example I will come back to in the disease mechanisms of certain microRNAs, and that's about microRNA 2. So, as opposed to microRNAs, siRNAs can have many origins, and most of them are depicted here. So you can have endogenous transcripts, which even may derive from transposons, so very old genes that have originated from other organisms. Then you have viral RNAs that can be introducing these siRNAs. Hairpin RNAs, I'll come back to that later. These are RNAs that you usually introduce artificially. You can clone them into a plasmid. This plasmid can be expressed by a virus, and then you have an ideal tool to use in the lab to introduce a short hairpin RNA into a cell of interest to study a process or a gene of interest. So this links to the environmentally, experimentally, or clinically introduced double-stranded RNAs, which are a shortcut to avoid having to use viruses that introduce hairpin RNAs. You cannot directly inject double-stranded RNA into a cell. The disadvantage is that you immediately have RNA that is very prone to degradation. You have endogenous, what's becoming more and more apparent is what I said before, that genes encode for inhibitors within their own sequence that regulate their own expression. So that's these negative feedback loops or positive feedback loops that become more and more known. It's actually quite an exciting field, and I think only the tip of the iceberg has been discovered now. So lots of for you to discover in the future. Transgene transcripts, that's for example if you want to make a transgenic mouse, you can also use siRNA as a tool, and RNAs which I don't know actually. Not important. So now we know all this, we can think about, or we can try to interpret the pathways that siRNAs and microRNAs use to achieve their goal. And the ultimate goal is, of course, the inhibition of gene expression, or the inhibition of protein expression, I would say. And both pathways use similar tools to get there, similar protein complexes. The end goal is also similar, so protein expression is prevented, but siRNAs, or double-stranded RNAs, have as end product the degradation of the messenger RNA target, while the microRNAs have as end product the translational repression of their messenger RNA targets. Exactly what happens to these messenger RNA targets of microRNAs, we don't know yet. Some people think they are stored within, what's that name again? Some sort of vesicles in the cell, which I don't really believe in. What's the name? P-bodies. Very good. Thank you. So some people think they are stored temporarily into P-bodies within the cell, and then when later signaling says that they have to be used again, these messenger RNAs presumably are released from the microRNAs and then enter the translation machinery. I wouldn't really understand how this would work because you need some sort of very specific signal, and mostly these microRNAs are quite small and are not really suitable to transduce specific signals. Understand what I mean? So the start of the CRNA pathway, I'll come back later to where it comes from. You have double-stranded RNA, and in the case of the microRNA pathway, you start from genomic DNA, which is transcribed by just the normal RNA polymerase II into microRNA, which has a poly-A tail as well. So a normal gene, which is then processed into this short hairpin loop by drosha, so just the tails are cut off. Drosha is an RNase 3 enzyme, and then exported from the nucleus to the cytoplasm by exportin 5. So this is quite important. Then in the cytoplasm you have this free microRNA hairpin that is then first processed. And from then on, the processes that lead to the incorporation of the short RNA molecule into the RISC complex are similar for siRNA pathway and for MENA pathway. So first, the hairpin is diced off. We call it dicing, and the enzyme that does it is also an RNase 3 enzyme, which is called dicing. Easy to remember. So then, the hairpin is cut off, and the double-stranded RNA is incorporated in the RISC complex. And the RISC stands for RNA inducing silencing complex. It's a protein complex that consists of several proteins, including, for example, organelle 2, which is often used in the lab as a method to study the RISC complex functionally. So both the siRNA and the microRNA are incorporated in this RISC complex. They are unwound, so one RNA strand is selected, and that is the guide strand. The guide strand is incorporated or remains incorporated in the RISC complex and will start searching for the target messenger RNA. The other strand, it's the passenger strand, is presumably degraded, but that would also sound like a waste of biological energy, cellular energy. And what we know now from, at least from microRNAs, is that often this passenger strand is also used in another siRNA or in another RISC complex. By mechanisms that are not entirely clear yet, this RISC complex, or protein complex with the small RNA inside of it, starts looking for messenger RNAs that are complementary, or at least partly complementary. In the case of siRNA, they will be fully complementary, and that's why the double-stranded RNA that is formed between the siRNA and the target messenger RNA is perfectly complementary and thereby is degraded, because double-stranded RNA in a cell is not tolerated. That's the whole principle of RNA. And the siRNA is reused. No. These are slides from someone else. So the siRNA is not reused. I don't know why that arrow goes up here. In the case of the microRNA, the binding between the microRNA and the messenger RNA will mostly not be fully complementary. We call it, there will always be a wobble binding phase pairing inside. And that causes the inhibition not to be pulled. So that means that the duplex is not fully double-stranded, so it's not degraded in most of the cases, and only the translation will be repressed. What nowadays we know that somehow these messenger RNAs are still degraded, by which mechanism we don't understand yet. So back to the slide on the overview, I will now go into the source. And here you can see that while the microRNAs are endogenous, this is the cell nucleus, double-stranded RNAs are often synthetic. So you can introduce synthetic siRNA. You can introduce short heptan RNA vector into the nucleus, which is incorporated, you do it well, it's incorporated into the genome of the host cell and then produces somehow endogenous short heptan RNA, which will then be entering the risk complex and probably function like that because that's the way you designed it. So function to degrade the messenger RNA. And how does it do that? So first possibility is indeed the degradation of messenger RNA. That's sequence-directed cleavage by endonuclease. So the double-stranded RNA is cleaved and cut exactly in the middle. Here, and there is perfect complementarity between siRNA and messenger RNA. And this happens predominantly in plants. For microRNAs, it always happens in plants and almost never happens in humans or in higher animals. And for siRNAs, it happens in plants and animals. It takes place in the cytoplasm. Then there is a sequence-directed inhibition of protein production. We call the risk complex in this case, and it's a microRNA and there is no full complementarity. Then we call this also the microRNA MER-NP. So that stands for microRNA. That's a good question. Look that. It's such a common term that I forgot the abbreviation. Anyway, so it inhibits the translation from start. So the DNA polymerase, or the... Sorry, I'm completely confused. Anyway, it takes place in the cytoplasm and it is enable root in mammals. Then there is the epigenetic, the epigenetic role of small interfering RNAs. That's sequence-directed inhibition of gene expression, and that works mainly on the chromosome. So it mainly works on the genomic DNA level to somehow manipulate the chromatin from an active state to an inactive state, and then inhibit transcription of genes. So the key question is to slice or not to slice. This is according to this Professor Zomers who gave me this slide. So once again, when you have perfect complementarity, you will have mRNA degradation. When you have partial complementarity, you will have translational repression. And this is mostly the case for microRNAs. So now I'll go more into the field that I'm more familiar with, the microRNAs. They actually add an extra dimension to gene expression regulation. So they fine-tune the expression of proteins. They do not fully inhibit the expression of proteins, but they fine-tune like 20 to 30% of the expression of proteins, so that cellular processes can be directed in a certain direction, can be finely modulated. It's not an on-off effect. MicroRNAs do not have an on-off effect. They have a very subtle effect on protein expression. They can also have multiple targets, and people think that they have these targets in similar pathways. That's not fully proven yet. We have a lot of things to do because there is now 1,000 microRNAs in the genome, and lots of them have different functions in different cell types, depending on the co-expression of their targets. The target prediction programs are not very good yet because it's difficult. You can understand that the partial complementarity between the microRNA and its target will also make it very difficult to predict which targets will be a target of microRNA. So in that sense, it's not easy to get a full understanding of the repertoire, target repertoire of a microRNA in a certain cell type without doing large-scale pull-down assays and then doing microarray studies. And even still then, it's very, very difficult. So what I'm saying now is that we think microRNAs function to target messenger RNAs that have similar roles, so are involved in similar pathways, but actually the definite proof that has not been given for any single microRNA. It's just maybe to just make our lives easier, because reality is becoming more and more complicated the more we know, the less we know. What we do know about microRNAs is that they can have tissue-specific expression patterns. And what is nice is that specific microRNA inhibiting drugs are now being tested in phase 2 clinical trials to, for example, target hepatitis C. So what's been discovered is that there is a liver-specific microRNA, microRNA-122, that has an important role in the replication of the hepatitis C virus. And because it's so liver-specific, inhibiting drugs of this microRNA will not affect the functioning of other tissues. And that's a major advantage above other drugs targeting this virus. And so the entomers that inhibit the role of microRNA-122 are really, really successful in inhibiting the replication of the hepatitis C virus. And another interesting thing of this story is that microRNA-122 is used by this virus as an endogenous gene to enhance the replication of the virus. Actually, this means that viruses are much smarter than higher organisms in trying to survive. So as I said, microRNAs regulate the expression of entire gene networks in health and disease, and these targets can often have functionally related roles. MicroRNAs can also have tissue-specific expression patterns, like I said, microRNA-122, which is actually not part of this slide, but there are others, like microRNA-133, which is only expressed in muscle, so in skeletal muscle and in the heart. Another example of cardiac-specific expression is microRNA-1, here. I think. So microRNA-1 and microRNA-208 are also very specific for the heart. They are expressed, so microRNA-208, for example, is expressed within the myosin heavy chain gene and also regulates the myosin heavy chain gene. I will come back to later. Actually, the heart is one of the few organs that has very, very specific microRNAs. And these microRNAs are also used in the clinic at the moment, or at least use is started for diagnostics. Because you can imagine if you have a heart infarct, a myocardial infarction, then cells are disrupted and their content is freed into the circulation, and microRNAs will also be freed. So the existence of, or the presence of cardiac-specific microRNAs in the circulation directly relates to cardiac damage, which is now being optimized as a clinical diagnostic marker for myocardial infarction. This slide is just to show to you that microRNAs have a lot of roles in development. So when microRNAs were discovered, they were first studied in lower organisms like C. elegans and in Arabidopsis in plants. And then the first thing that researchers do is look at their role in development. When genes have a role in development, they are often regarded as very important for the function of the organism. Secondly, what's being done then is normally the investigation of the role of these genes, of novel genes in cancer. And the other fields like heart come a bit later. So the role of a microRNA in the development of the heart, one of the first things that was published was only in 2007. That was the negative feedback loop of the microRNA-1 and 133 expression on their own expression. So microRNA 133 and 1 are encoded within the same gene, so they are polycystronic. They have both the same promoter, which involves the SRF binding site. SRF stands for serum response factor. It's a very important transcription factor in not only the heart but also in other systems. And the serum response factor normally drives the expression of these two microRNAs. But you can see that one microRNA has an important role in myocyte differentiation, while the other, the second, 133, has an important role in myocyte proliferation. And these are opposite processes, and they should be balanced. And somehow this gene regulates itself. So microRNA 133 directly targets the expression of and activity of the SRF transcription factors, and thereby downregulates its own expression. This is a way for the heart to somehow balance between differentiation of myocytes and proliferation of myocytes, which is very important in the development of an organism, because initially myocytes, the heart needs to grow, so proliferation is more important, and in a later stage these myocytes need to develop into a more adult phenotype, and then differentiation is more important. We now also know that these patterns are regional, so they differ in regions. We don't exactly understand yet how the cell knows whether microRNA 1 or microRNA 133 should be prominent, but this is also the subject of current investigation. And I wanted to show you this because it's a very good example of a negative feedback loop that is mediated by microRNAs, and it's quite simple. So what about the role of microRNAs in disease? In disease you have stress signals, very general. So a cell encounters a stress signal that is being introduced by a pathogen, a virus, a bacteria, or by cancer cells. Cancer is also stress. Or, for example, in terms of the heart, it's introduced by high blood pressure or an infarction. Signal transduction pathways are activated, and some microRNAs go up and some microRNAs go down. It's just a normal gene transcription mechanism. They also have their own transcription factors, as I said before, so they will be regulated on the transcription number, or on the transcription level, to modify the number of microRNAs that are present in the cell. So on the one hand, an increase in microRNA number, you can see on the right, will result in an increase in depression of targets, so a decreased expression of messenger RNA. And on the other hand, a decrease in microRNA number will lead to an increase in the target messenger RNAs. So this is quite an opposite way of thinking. So the cause of abnormal microRNA expression can be the altered expression regulation by transcription factors, as I said before. For example, when in disease SRF transcription factor is affected, then also the downstream microRNAs will be affected and the whole feedback loop will be modified. But in cancer, there is also other possibilities. Not so much in the heart, as far as we know now. In cancer, you can, of course, have the chromosomal abnormalities, microRNAs in genomic regions prone to alterations in cancer, shifts or replications, amplification of regions within oncogenes. That's the word I was looking for in the beginning of the lecture. Oncogenes and tumor suppressor genes. Anyway, we see a lot of cancer. Epigenetic changes, 50% of the microRNAs are associated with CpG elements, and as you will know, CpG elements are associated with gene expression. So then microRNAs are candidate targets of the DNA methylation machinery. And then, of course, there is the possibility of mutations and polymorphisms in microRNAs that affect either the processing or the levels of certain microRNAs. So the processing or the function of certain microRNAs. In reality, we see that in cancer, indeed, some microRNAs are located within mutational hotspots, we call them. In heart failure and cardiac disease, that's less prominent. We have actually done a lot of work in sequencing microRNAs in heart failure patients. and never found one single mutation. This means that these microRNAs, there is quite a restriction on their mutation capacity, let's call it that way. So there is a high control of their functioning. And you can also imagine why that is, because normally when you have protein coding genes, a codon, an amino acid, is determined by 3 nucleic acids. In microRNA case, you have a one-on-one relationship. Every single nucleotide is important for the functioning of microRNA. And probably that's the explanation that we always give for their very high conservation and also very low mutation rates in normal populations. Related to normal populations like patients. Cancer is often a disease that develops earlier in life, while heart failure is often a disease that develops like late in life. Unless it's hereditary, but so far also in hereditary forms of heart failure, no mutations have been found in microRNAs. And then there can be defects in the microRNA biogenesis machinery. For example, dicer can be mutated. However, that's not very likely because when we, so in the lab, people have studied the role of these enzymes, and when you just knock out dicer, for example, animals will not, they will develop heart failure. When you get dicer specifically out of the cardiomyocyte, so that's a very specific deletion in the cardiomyocyte, mice will survive development, but will die shortly after from ventricular septal defects. When you, another tool is to make an inducible cardiac-specific knockout. We've done that as well. And what you then see is that when you just induce the deletion of dicer in the cardiomyocyte in a later stage of life, mice will also spontaneously develop. So this is just to illustrate the very central and important roles of these enzymes in development and organism function, and therefore I find it highly likely that these biogenesis machinery proteins will be the source of abnormal microRNA expression. But we do know that during heart failure, for example, dicer expression is increased. Exactly how that is caused, we don't know. So what about microRNAs in cardiac disease? In 2006, the first studies were published. And what people typically do is they look at gene expression arrays. For microRNAs, that was not that easy. I don't know if you were involved in the course on gene environment interactions, 0.5, 0.6, nanotechnology. So I already gave a lecture on microRNAs. No? Right. Oh, then it's on. So what people typically do when they want to know more about the role of certain genes in the disease is they do a microRNA study. Do you know what microRNAs are? Okay, so what I want to say here is just compare a healthy heart with a heart that is prone to develop heart failure. And a heart that is prone to develop heart failure, in my case, is a heart that's been subjected to pressure overload, to high blood pressure. And typically, a heart muscle thickens to increase the power pump against the increased pressure in the aorta. That's typically the response of a heart to pressure overload, to hypertension. However, you can imagine that this condition of the thickened heart muscle is beneficial in the beginning because the heart can generate more power. It's like a bodybuilder. At the end, it will become detrimental because the muscle mass will become its own inhibitor. It will get in the way. A bodybuilder cannot run very fast because it's too heavy, it's too immobile. So we want to visualize what happens in terms of microRNA expression at the molecular level in these hearts. So the first study, as I said, in 2006 by a colleague, Eva van Looy, in in Dallas, and she's in a very, very good lab. They were the first to study the role of microRNAs in the heart and in heart failure, and they found a lot of them that were regulated in certain models of hypertrophy, overload in heart. This is a typical way of visualizing the gene expression array. You look at, they looked at two different models of hypertrophy, heart failure, and then they looked at the overlap between these models in terms of upregulated microRNAs and downregulated microRNAs. And they selected two of them, all both are upregulated. One of them is 195, and the other one is microRNA 214. In the meantime, other labs have also addressed the role of microRNA 221 and 23A, 93, 133A, 150. So a lot of studies have in the meantime been done, but they were the first to discover these microRNAs for their role in heart failure. They confirmed this by non-blotting, which is very important when you do microRNA array studies. The limitation is that the microRNAs are only very short, so you have not a very good choice of the technique. So you cannot really say, you cannot really determine the hybridization temperature of the probe. I do not understand what I'm trying to say. Yeah, so they are very short. You have no choice in nucleic acids that you are going to use for your probe. You just have to use the full-length microRNA probe complementary to the microRNA. And then you don't have a very good choice of the hybridization temperature. The hybridization temperature will be all over the place, the optimal, and you can only use it for an array if you want temperature. That means that your aspecificity is going up. And you use only your low temperature to make sure that all the microRNAs find that complementary probe. And then that increases the aspecificity of the approach. So therefore, for microRNA arrays, it is very important to confirm the findings by non-blot. That's an independent technique. And that's what they did. And then they focused also on the role of microRNA 208 in a later study. So I first want to tell something about 195. So they found it by this microRNA array study, and they just make it transgenic. In America, they're very easy with that. They find the gene and they immediately go from the discovery into making a mouse model. So they made it transgenic, made actually 3 lines. And one line had a very high expression of this microRNA, you can see it here. And immediately these mice developed heart failure. You can actually see this is a 2-week-old mouse. This is a normal heart, which has a normal wall. So this is the left ventricle here, septum, and the right ventricle here. Atria on top. You can see that this transgenic mouse has a huge atria and has not much of a cardiac wall. So you can see that this single microRNA has quite a dramatic effect only in cardiomyocytes. This is a cardiomyocyte-specific transgenic animal. You can imagine that this line died really quickly. And then there was another line that had a bit more modest expression of overexpression of this microRNA. And these only developed heart failure in a later stage, but similar as the line 3. Also have huge atria and very thin ventricles. And a lot of cells that actually do not belong in a normal heart. These are the blue cells you can see at the bottom. They made another transgenic, and just to add that this was the microRNA 214 that they also found upregulated. And here you see that these microRNA, or these transgenics, do not have a phenotype at all. So just to illustrate to you that success is not guaranteed. Not all microRNAs that are upregulated have very central roles in the development of heart. So then there is this microRNA 208 that I've already been talking about, which is restricted to the heart. It is encoded within the entrance of the genes that encode for the 2 myosin heavy chains. So microRNA 208A is encoded within the myosin heavy chain 6 gene, which is also the alpha myosin heavy chain gene. And microRNA 208B is encoded within the beta gene, that's the myosin heavy chain 7. You can see that the conservation, which always strikes people, is quite high. So you have the introns here in the blue boxes, the introns of the myosin heavy chain gene, and you can see that the conservation is very good. But then somehow this mysterious conservation that before the discovery of microRNAs nobody understood. So nobody understood why actually this is even better conserved than on average the introns of the host gene. So now we do understand because this is the microRNA. They made knockouts for this microRNA. No need to do it cardiac specific because it's already a cardiac specific microRNA. So they made just full body knockouts. And at baseline they did not have a phenotype, so they were quite disappointed. And then they studied the influence of pressure overload on these knockout mice. And then they saw that while in the wild type, there is a normal response to pressure overload, the heart becomes bigger, the walls become bigger, there is some interstitial fibrosis to keep the muscle cells together. And none of this happened in the knockouts. So what they found was that during stress, so during pressure overload in the mouse, the stress signaling normally activates the thyroid receptor and thereby activates alpha myosin heavy chain expression, which is dependent on thyroid receptor signaling. And this expression is normally increased, and thereby it also increases microRNA-208 expression, because they said, these authors, so also Evil and Boyd, said that the microRNA-208 expression is equal, or it has the same promoter as the host gene, so as the myosin heavy chain gene. And then they designed this beautiful feedback loop again, which is a bit more complicated than the one I showed before. And I think it's quite inventive. So you have the beta myosin heavy chain gene, which encodes microRNA-208B, which they don't mention, and the alpha myosin heavy chain gene, which encodes microRNA-208A, which then inhibits certain targets that are not important at the moment, which can direct thyroid hormone receptor signaling to inhibit the expression of beta myosin heavy chain. And somehow they say that the switch that happens during heart failure in the contractile types. So you have the beta myosin heavy chain that directs slow contraction. Do you know that? And the alpha myosin heavy chain that directs fast contraction. Somehow these microRNAs help to fine-tune again, to modulate the balance between the alpha and the beta myosin heavy chain. And this is quite a very important finding. Very central in the development of heart failure. So as I said before, the deletion of DICE on the heart leads to rapidly progressive dilated cardiomyopathy, heart failure, and postnatal death. And that is by the misexpression of cardiac contractile proteins, the sarcomere disarray, and apoptosis that is seen in these hearts when DICE is knocked out. And this was actually published by Paula Costa Martens and another group, with Paula Costa Martens is part of the cardiology department in Maastricht. So you can see that in Maastricht we are not that bad at doing microRNA research. In conclusion, there are several specific microRNAs that regulate cardiac development and hypertrophy. And the challenge always is finding the mechanism. So one microRNA, as I said, can fine-tune the expression of several target genes. Although I've shown you now very simple feedback mechanisms, probably this microRNA, you know that microRNA-208, for example, has many more targets than only this thyroid receptor hormone signaling. So the key is to find other ways in which microRNA-208 modulates the expression of heart failure. The relative contribution of all these leads to the observed phenotype. So again, we only know the tip of the iceberg. Our interest is the role of inflammation-related microRNAs in cardiac pathology within my lab. First, before I go there, before the break also, I would like to quickly go over the use of RNAi as an experimental tool in research, so that you can understand better what I'll be talking about in my practical part after the break. What do you want to break down? Continue? So, as I said before, the experimental source of RNAi can be multiple. So this is just a repeat of the previous slide. You can use short hairpin RNAs in a vector. You can just order from a company synthetic small interfering RNAs. You can, for microRNAs, you can now order from a company directly anti-MERS inhibiting microRNA function, and you can order pre-MERS, pre-mimics, they call it, to mimic. So to go in a little bit more detail, what you can do in animals is introduce long double-stranded RNA by microinjection, for example, and that's probably mostly done in vitro in cell cultures. And then this long double-stranded RNA will be processed by Dicer into a protocol and will immediately be incorporated into a RISC complex. Synthetic siRNA can be directly microinjected in the cell. The disadvantage is that it's RNA, and that's for both of them actually. And the RNA is quickly degraded, and that's not very useful for in vivo use. Because you can imagine that if you inject an RNA into the circulation, it will be immediately degraded by circulating proteins. They're very active in degradation of products that do not belong in circulation. So that also immediately inhibits the use of RNAs for therapeutic goals. You can use plasmids to encode short hairpin RNA behind the promoter. And you can introduce this vector into a virus, which makes it even better, because then you can use the characteristics of the virus to get the short hairpin RNA inside the cell. And then you just undress the virus so that it's not replicated anymore, so it's not damaged anymore. Inside the cell, so if you use the latter two approaches here, the product needs to be produced, so it needs to go to the nucleus, short hairpin RNA is formed, and by exportin 5 is introduced into the RISC complex. And eventually it follows the whole Dicer machinery. So siRNA versus short hairpin RNA. siRNA is synthetic. As I said, short hairpin RNA is always vector-based. The advantages of siRNA are that it's synthetic, so it's ready to bind. You can be very quick. You can control the dose very easily, and you can chemically modify them. So you can make them more stable, or you can label them somehow so you can follow them inside the cell. And you can easily pull them as well to target the whole length of a gene, for example. You can use large DNA to make transfection very efficient, but the target cells are limited, so you can only do this in cell lines, for example, that are rapidly dividing, that are very accessible to foreign products. You cannot use or barely use this strategy in cardiomyocytes because they are terminally differentiated. They do not allow products to come in this way. Barely allowed. You knock down the strangent, so the advantage of a cell line is immediately also the disadvantage, because when you wait too long, your effect will be diluted. And you cannot renew it, so you always have to do it again, which damages the cell and will shorten its survival. But siRNA is nice, but you have to be able to really do it quick, and then have short-term readouts. The advantages of short hairpin RNA inside a virus, for example, are that you can always do it again. You can do a stable knockdown by, for example, using an antiviral vector basis. Antivirus is incorporated into the cellular boost, into the cellular genome, and then it doesn't matter how often the cell replicates, it'll still be there. There's no dilution of the You can do this in vivo as well, and I'll come back to that in the examples I'll give you. The disadvantage is that the vector construction takes some time. You need to have a lot of knowledge inside your lab. You cannot combine. It's more difficult to combine, for example, to pull several RNA molecules to target one single gene. And the design rules of these factors are not really well understood. So you just go with what you get in the company, but actually you never know what it works. Potential issues of in vivo use of sRNAs is that all sRNAs can elicit immune response, which is, of course, when you think in terms of therapy, it's not very desirable. Then your disease needs to be so severe that outbalance the disadvantages of an immune response. Then the functional delivery of sRNAs in vivo is challenging. Like I said before, you cannot prevent the degradation of RNA when you inject it in the mouse, let alone when you have a pill that is ingested by humans and into the stomach. Already immediately degraded. Then you have to get it inside the cell. Somehow the cell needs to open up for this foreign material, which it will, by nature, of course, not do. And then, when you have overcome all these hurdles, you have to get it inside the RISC complex. And actually, when you've already formed RNA structures, that appears to be a hurdle that cannot be taken. So that's the reason why siRNA therapy is built, gene therapy is built so far. Then there's a lot of specificity issues, off-target effects, and you can also have toxic effects, endogenous RNA pathway. So the siRNA that you introduce will, of course, use the same pathways to be processed, and the endogenous microRNAs, for example. And that can be a very severe side effect as well, which you simply, by injecting a lot of RNA to produce enough to reach your therapeutic potential, also overload your endogenous machinery. So that's a major disadvantage, which is also true for SHRNA. So more recent experimental use is the use of antimers. You can use them both in vivo and in vitro. In vivo is easy because antimers can be DNA. The only thing they need to do is enter the cell. And so they need to survive the circulation and enter the cell. DNA molecules are better at that than RNA molecules. And inside the cell, the only thing they need to do is find their complementary microRNA. So that does not depend, the function does not depend on the incorporation into a RISC complex. And that is very important, very handy. In vivo primers can be introduced by viral vectors. In vitro, they can also be just injected, but then these primers need to be RNA. So in vitro, that's easier. You can just get them inside the cell. You can have them, I specifically say, primers, so they still have a hairpin that needs to be processed, and then they will immediately or automatically be incorporated in this pathway that will get them into a RISC and get them through. That was it. I don't know if there's any questions for this first part. So shall we have a 10 minutes break, and for those who are interested, you can come back to the second part. It will only be short. It will only be 20 minutes on the studies that we do in our lab. And, the use of these, all the different terms that I've just introduced to you. Which will make them a bit more alive. So can you come back at 5 minutes?